Alright, this is part 2 for the multiple choice parts of your test study guide, Exam 2. So the next one I want to talk about is composition of a phospholipid. I also talked about this in your short answer questions. But basically you have a hydrophilic head and a hydrophobic tail. Your hydrophilic head faces outwards and the hydrophobic tails face inwards in your phospholipid bilayer. The head will touch the water parts, the tails will look at themselves because they don't like water. Exergonic and endergonic reactions, which ones produce or use ATP? Exergonic reactions are reactions that produce energy. So they release energy, they release or make ATP. Endergonic reactions are ones that require energy to be put into the reaction in order for the reaction to take place. So they would use the ATP. Potential versus kinetic energy examples. You have a couple of questions on your test. One of them talks about a battery. A battery is always potential energy because it's energy that's stored and hasn't been used yet. And you have some that ask you to identify kinetic energy. I'm trying to find it again. I've ran through these. Let me just look up the word kinetic in my nice little list. Oh, nope, it doesn't have what I was thinking of. But kinetic energy is the energy of movement or motion. Potential energy is the energy that is stored on one point or the other. So depending on what you're looking for, or what question you're looking at, you need to know those two terms. Proteins, three-dimensional shape, structure to function. I talked about this again in the short answers, but basically the shape of the protein, the shape of your enzyme, has to match the shape of the substrate in order for the reaction to take place. If anything that happens to that shape, it will no longer work because they are very specific. Each protein enzyme is specific to its substrate. Other facts you need to know about enzymes while we're talking about enzymes are enzymes allow reactions to occur at a moderate temperature. You don't have to heat things up a whole bunch. Enzymes are reusable, so they can be used over and over and over again. They bind to their substrates in such a way that the reaction can occur more readily. So they kind of get the parts of the substrate that need to bond to each other facing in the right direction. They lower the energy of activation for a reaction. So they reduce the energy that's needed to be input in the system for the reaction to occur. And they'll bring together a couple of molecules or a few molecules and cause them to interact with one another. So they are a point of lost a word, but like if a bunch of people were coming together into one room so that they could have a meeting, that room is the area for the meeting that represents the enzyme. It's just the location that the reaction can happen. The place where an enzyme where substrates fit in such a way they are oriented to react is called the active site. So the place where the enzymes will react with the substrate is the active site, and again, that active site is very specific. One enzyme per reaction. Only certain substrates can join in. Now, in allosteric regulators, what happens is you have a spot on your enzyme that's not the like this is my active site. You have another spot on your enzyme where a regulator can come in and it can attach, and it will either inhibit or increase your reaction. It can go either way. But sometimes if a phosphate group is added, it might increase the ability for that enzyme to react. It might change the active site in some way or just allow it to work a little better. If it's an inhibitor, maybe the substrate can still bond but to the enzyme, but the substrate can't bond to each other. It no longer can do its reaction. So it just depends on which type of enzyme or which type of allosteric area it is, but that's what's happening in the allosteric regulators. Is there's another spot that's something that's not a protein can bond to change it. Activation energy is the energy required for the reaction to take place, and enzymes lower that. Factors which affect functionality: temperature. So if an enzyme gets too hot, it can't work. If an enzyme gets too cold, it can't work. If the pH of an enzyme changes, it also can't work. Those are the few main things. Lock and key model, I just talked about that. One enzyme to one reaction. Coupled reactions. In a coupled reaction, your exergonic reaction is going to fuel your endergonic reaction. So think the stages of photosynthesis, which we talked about this week in lab. So your first reaction is your light reaction in photosynthesis. So in that reaction, you are taking sunlight and you are making chemical energy. And from that chemical energy that you make, you are going to build glucose molecules. So you start with an exergonic reaction, but then you are building something with endergonic. The cell doesn't actually make energy for itself in these two reactions, but part one fuels part two. So exergonic fuels endergonic. They are coupled reactions that is taking place. That's a good example, great example is photosynthesis. However, that example is not on your test, but just to get you thinking in the right direction. And I have catalysts on your study guide because enzymes are also known as catalysts. That's one of the big things. I skipped ahead, I think. Proto-oncogenes versus oncogenes. You have a question on your test. Oh, maybe not. About these. But proto-oncogenes are genes that regulate your cell cycle. I'm just going to go ahead and talk about it instead of read the actual question. Too bad. But proto-oncogenes regulate the cell cycle. They are ones that are responsible for controlling the how quickly they divide and what happens during cell division and all that stuff. They are not necessarily cancer-causing, but they can at some point get mutated, and then once they do mutate, they become oncogenes, and those are what causes your tumors, and those are what causes your cancers, because at this point, your cell doesn't know to quit dividing. It doesn't know to go into apoptosis and die. It just will divide and divide and divide and divide. Semi-permeability of the cell membrane, we talked about that. So I believe that wraps us up pretty much with everything that you need for your multiple choice parts of your test. I'm going to quickly just read through here. Make sure that I covered everything that I wanted to cover. Yep. I like all that stuff. Oh, one more thing. Anytime you are releasing energy in a reaction, you are doing your second law of thermodynamics, which means you are taking one form of energy, expelling it, and when you release that energy, it's breaking apart into different forms, and you oftentimes lose energy as heat. That's your second law of thermodynamics, and that will also show up as a question on your test. And that's it. So I hope these are helpful.